Hello, everybody. So we're going to start on chapter 2 today. We finished up with chapter 1 where we talked about the action potential. We're going to go through that again when we meet in person again. We're going to practice that a little bit. But for this recording, which was supposed to be Monday's lecture, I'm going to start into chapter 2 where we start talking about the synapse and chemical communication. I'm not going to do the full thing, just as much as I really want to talk about until we can meet again and kind of revisit that material as well. Okay, so let me get the PowerPoint going. Okay, so synapses, so far we've talked about electrical happenings, the action potential, changes in ions going in and out, causing membrane potential changes. If we reach threshold of excitation, then it fires an action potential. So today we're going to zoom in on the synapses when that action potential reaches the terminal buttons and releases neurotransmitters. So the synapse is considered the terminal button, the space in between the terminal buttons and the dendrites or soma, right? And the neurotransmitters are leaving the terminal button into the synaptic space, or we call it the synaptic cleft, binding to dendrites or soma. Most of the time it's going to be dendrites, releasing, and then either getting recycled or destroyed. Most of the time it gets recycled and repackaged. So we're just going to talk a little bit about the synapse today, what these guys are, EPSPs and IPSPs, and then we'll save neurotransmitters for next time. Okay, so the way that it was discovered that we even had something called a synapse, because it could have just been electrical communication, and in fact in invertebrates, a lot of the neuron communication doesn't involve chemistry at all. It just goes electrically the entire way down between neurons, and there's no neurotransmitters involved. But in vertebrates and higher mammals, well, I just should say vertebrates, and especially in humans, pretty much every single one of our neurons, when they want to communicate with another neuron, it has to involve a chemical component. So the way this was discovered was Charles Sherrington in the picture here. He was measuring how fast two neurons were communicating with one another. We at this point knew the speed of conduction, how fast an action potential was. We believed it was probably the speed of light because it was so fast, because you can do everything at the same time and so quickly that like you're doing it before you can even think about it. But until it was measured and objectively found, it's not the speed of light, it's a lot slower than that. It's still really, really fast. We could do proper experimentation and see, okay, do different neurons have different speeds? And what happens when neurons are communicating? So Sherrington was looking at the reflex arc. I believe we had talked about this when we were talking about sensory neurons, motor neurons, and then interneurons, and this is what you can see here. I hope my little mouse is showing up. Sometimes I blow this up to a big red mouse, but I forgot to do that today. But here on your skin, you might touch something. A sensory neuron sends that information, right, from your hands to your spinal cord. In the spinal cord, here it's labeling as intrinsic neuron. Interneuron's the more appropriate term, but a little interneuron is basically connecting the sensory to the motor neuron, and then the motor neuron comes out of the spinal cord and goes to, I guess we were talking about your knee, so the patellar reflex, not touching with your fingers, but nevertheless, that is the simplest circuit that we can build, sensory to inter to motor neuron. So Sherrington was looking at these reflexes and trying to measure how quickly these were happening. Pretty much the fastest human reflex that we have is about 50 milliseconds. For example, your blink reflex, it's fast, right? But it does take some time because that communication has to happen between neurons. So the speed of conduction along an axon, again, this really varies. It varies by species and where we're looking at, but ballpark will go with 40 meters per second. Very, very, very fast. But what Sherrington noticed, is that if you're measuring that speed of conduction through a reflex arc where multiple neurons are involved, it seems to be a lot slower. It was 15 meters per second. So something slowed down the communication. And so he was basically proposing, oh, I forgot I left this. Yeah. Actually, I think we talked about this, that reflexes don't require the central nervous system, but we will come back to this, especially when we do neuroanatomy. So back to this idea that the speed wasn't as fast as we measured in a single axon. Basically, Sherrington proposed something else has to be happening here. And what that other thing that is happening is that potentially, and again, this is back in Sherrington's time, now we know what that is, but he said potentially a different form of communication is happening, not electricity. And now we know it's neurotransmitters, and that takes a little bit of extra time for those neurotransmitters to work. So, our neuron structure revisited real quick, our dendrites, right? Our dendrites are getting input from other neurons. Neurotransmitters are binding to dendrites and causing changes there. Or a synapse is happening at the soma. The nucleus contains your DNA, where transcription and translation can occur. The axon, along the axon potential flows down. Axon collateral is if we have a split. The axon hillock is where the action potential starts, with nice fatty myelin wrapping around in little chunks and segments to help protect the axon. Right, if we're in the central nervous system, these will be oligodendrocytes. One oligodendrocyte, which is a glial cell, wrapping around multiple sections, potentially multiple axons. Whereas if we're in the peripheral nervous system, then we're talking about Schwann cells. And with a Schwann cell, every single glial cell is a Schwann cell. Every single segment of myelin sheath is one Schwann cell. In between the nodes of Ranvier, this is where speed boosters happen for the action potential. The axon terminals are the terminal buttons, and inside the terminal buttons are the neurotransmitters. So these are chemical, different structures. A lot of them are amino acids, but nevertheless, they're molecules, they're chemicals that are released from the neurotransmitters, cross the synaptic space, and go and bind. And that's what these things are showing here. These little like pinchy things. This is where the neurotransmitter attaches to a receptor, which is going to be an ion channel. The neurotransmitter attaches to it. It fits like a lock and key. Only serotonin is going to fit a serotonin receptor, which then opens up an ion channel, and then it releases and goes back to the terminal button. And these neurotransmitters are either excitatory or inhibitory. We'll get back to that when we talk much more explicitly about the neurotransmitters, the different types, and kind of how they work. So dendritic spines, right? So on the dendrites are where we have synapses with other neurons occurring. What's really cool about it is that some of these dendrites, right? So these kind of like tree formations that are coming off the cell body, some of them appear to be bumpy, and now we've uncovered that this bumpiness on the dendrites, we call them dendritic spines. This is where active learning is occurring. So if you are learning something over several days and weeks, a dendrite is not just going to sit here. It's kind of going to grow out a tiny little bump and go and connect with the terminal buttons of another neuron. So the more you use those connections, you use those synapses, those spines get thicker and grow out more. And we see this almost in real time, where kind of when you are learning or forgetting or making new connections, especially in a developing child where they're making all of those connections, those spines are indicative of the learning that is occurring. More spines, meaning a neuron that is making a lot of connections, and ideally will translate later into long-term memory. Okay, so coming back to the synapse, right? The synapse includes the terminal button, the synaptic cleft, which is basically that space in between where those neurotransmitters are. When you guys were sketching your neurons, you drew little like dots or bubbles in between the dendrites and the terminal buttons. So that's the space where the neurotransmitters and ions are going to be hanging out, causing changes, and then going back into the terminal button. This guy right here might be a dendrite or might be a dendritic spine, right? Kind of like a protrusion off of the dendrite. Or this could be soma. A synapse can also happen directly on the cell body of a neuron. But the specific terminology I want to start introducing here is not just terminal button, but the membrane of the terminal button is called the presynaptic membrane. It's presynaptic, meaning before the synapse, whereas the other side of it, the dendrites, are the postsynaptic membrane. A lot of things are happening at the membrane level, so that we're going to continue talking about it. But just keep this in mind, right? At the end of an axon, at the end of a neuron, right? You have dendrites, cell body, axon, terminal buttons. The terminal buttons are the presynaptic area that is connecting to another. I don't even know if this is showing up on the video, where it's going and connecting to another neuron, and those dendrites are then post-synapse, right? So on the other side of the synapse, when neurotransmitters cross and go and connect. These binding sites where those neurotransmitters are connecting to, we call them receptors, and we're going to find out these are actually ion channels. So neurotransmitters or drugs, these are called ligands. They have the capacity to bind to one of these receptors and change its configuration. Meaning if this is a receptor and a neurotransmitter comes and it binds to it, because it's an ion channel, if this is the correct neurotransmitter to the correct receptor, meaning serotonin to a serotonin receptor, it's going to bind to it, open it up, and allow ions to pass through. So the neurotransmitter is a ligand, meaning a ligand is a chemical, is a molecule that has the capability of changing the configuration of another molecule. In this case, a protein, an ion channel, with the neurotransmitter changing that configuration of the protein. And again, drugs can do this too. We can have drugs, not neurotransmitters, that go and will bind to those as well. For example, alcohol can bind to what are called GABA receptors and cause them to open up. It doesn't have to be the neurotransmitter. But what happens when the neurotransmitter binds, it causes not only the ion channel to open, ions will go through, but when ions are going in and out, just like the action potential, ions flowing in and out translates to a change in the membrane potential, right? When we talked about the action potential, that means we either depolarized, we got more positive, or we hyperpolarized, we went more into the negative, if we ended up adding either positive ions or negative ions, right? Depending on kind of how it's either going in and out. And we talked about that with the action potential. The same thing is going to happen here. If we have positive ions like sodium entering the postsynaptic membrane of a dendrite, it's going to cause a postsynaptic depolarization. So, ions going in and out because of neurotransmitters. Neurotransmitter opened up that channel. Now an ion is going to go in or out. Most of the time it's ions going in. Depending on their charge, if it's positive charge or negative charge, regardless, it's going to change the membrane potential and cause postsynaptic potentials. And basically we have two categories of these. Either it's excitatory, right? We have a positive charge coming in that depolarizes, meaning it excites the dendrite. These would be called excitatory postsynaptic potentials. It's a very long term, but excitatory, just like we talked about with the action potential, meaning depolarizing, more positive. Postsynaptic, because we're on the other side of the synapse where the dendrite or the soma is. And then potentials, we're changing the voltage. So EPSPs get us closer to the threshold of excitation. If you're like listening this while like doing dishes, because sometimes people like, you'll like watch it and then you'll like listen to it again, but you're like doing dishes or laundry or something. Now would be a really great time to stop what you're doing and just write that down. EPSPs at the dendrites will get you closer to the threshold of excitation, right? When we talked about and I'll repeat it again, when we get closer to the action potential, right, we first need to get to the threshold. Once we reach threshold, that whole cascade of events occurs. But we didn't talk about what leads to you getting closer to that threshold. This is what leads you to get closer to that threshold, EPSPs. Dendrites, EPSP here, EPSP here, EPSP here, right? You might have 5 EPSPs. All of a sudden, the soma receives all of those excitatory potentials and it hits negative 55, so it fires an action potential. So again, EPSPs get you closer to the threshold of excitation. Inhibitory is the opposite. It gets you further away from reaching threshold. So inhibitory, this might be like a negative ion that entered, like chloride. We haven't talked about it yet, but chloride is a negatively charged ion. If it enters, we hyperpolarize, so we become more negative. And in this case, we call these inhibitory postsynaptic potentials. So because we're going negative, we're actually getting further away from negative 55. We're going into like negative 80 or negative 90. So when you have IPSPs, you are less likely, you meaning the neuron is less likely to fire an action potential because you are getting further away from that threshold. So again, these EPSPs and IPSPs, they are not the action potential yet, but it gets you closer or it moves you further away. If we have enough EPSPs, maybe we reach that threshold. Maybe we have a combination. Maybe you have 3 EPSPs and 2 IPSPs, right? Then it just depends on how strong it is. It's not just 3 to 2. These could be 2 really, really strong IPSPs, then that might outweigh it. So let's talk about that a little bit more in a minute. I forgot I had this in here. Gap junction. So I said they're invertebrates that don't use neurotransmitters. They don't have that chemical portion of the neural communication, and it's just electricity. So there's no synaptic space. You can see here, there's really, we call it a gap junction, but there's no space for neurotransmitters to be released. It's just kind of a pathway for electricity to continue flowing. So there are some electrical synapses, right, that do not require or do not use chemical communication. Again, this is more common in invertebrates, less so for vertebrates. We do, I believe, humans have some gap junctions, but they're very, very, very tiny neurons somewhere in the periphery. I forget where it is. Okay, so this process of having EPSPs and IPSPs all talking to one neuron, we call this neural integration, right? If we are going to reach threshold of excitation, here I'm showing it as negative 50, then we add up all of those potentials. So here, this might be a little EPSP bump, right? This one might be 2 EPSP bumps. So temporal summation, meaning we had one and then we had another, it's getting us a little bit closer to threshold. Or we have 3, boom, 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 kind of in succession, right? One, then the other, and that potentiates or depolarizes the membrane, and in this case reaches threshold and we fire in the action potential. So when you have that delay of incoming EPSPs, we call this temporal summation, right? Let's just say one dendrite is getting 1 EPSP, another EPSP, another EPSP. Then it's like, all right, I'm depolarized enough, fire away, blur. But what we can also have, I'll show it on the next slide, is not just one dendrite. We might have one EPSP happening over here and another one over here. So if both of these are getting excitation, right, from neurotransmitters binding and opening up ion channels, if both of these are excited, then that's spatial summation. So 2 different areas of the neuron getting excited and adding up to fire an action potential. So here I have a better graph for that, but here that's what this is showing. Simultaneous EPSPs combined spatially. So 2 different locations, right? Maybe a dendrite or maybe directly on the soma, but at 2 different locations. This temporal one would be at the same location, but multiple times. This number 5 illustrates an IPSP, right? We hyperpolarize, we get further away from the threshold. Okay. And this kind of shows the difference that I was just talking about. Temporal, right? Several onto one in quick succession. If you guys remember when we talked about neural coding, this would be, right, if it's kind of slow, it's like 1, then 2, then 3. It's kind of signaling a weak stimulus, whereas if this temporal summation was very rapid, like every 2 milliseconds, like boom, 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 then it would be coding for a very strong stimulus, such as a bright light. Down here would be spatial summation, right? So inputs at different locations, but happening at the same time, adding up. Now keep in mind, most of the time, it's going to be a combination of spatial and temporal, right? As I told you guys, a single neuron usually has hundreds to thousands other neurons talking to it. So you're going to be getting inputs at different locations at different times. So just keep that in mind, that it's almost always going to be both temporal and spatial. And then if these are EPSPs, they add up, reach threshold, boom, fire the action potential. Okay. So formally, the definition for neural integration is the interaction of these excitatory and inhibitory synapses, or rather these excitatory and inhibitory postsynaptic potentials would be the more appropriate way to say that, on a particular neuron, right? So the summation, how all of these guys are interacting, will determine whether or not we fire an action potential. So EPSPs increase the likelihood that we actually fire an action potential, whereas IPSPs, because they are inhibitory, EPSPs for excitatory, IPSPs for inhibitory postsynaptic potentials, we get further away from the threshold. This decreases the likelihood that that neuron will fire an action potential. So this here in red is showing a whole bunch of EPSPs. They all add up, fire an action potential, whereas this one is illustrating in red excitatory, but in purple or blue inhibitory, right? So then you have this neural integration of both EPSPs and IPSPs to determine if we reach threshold or not. You can illustrate it like this as well, right? We have some that are causing here plus for excitatory depolarization, right? We have a little bit of a bump up, whereas we have one negative one. The neural integration is the interactions of all these guys. And then the action potential question mark is, do we reach threshold? If yes, then fire. If not, nothing happens. So these little action potentials spiking, right? A single one of those is an action potential. It's often illustrated as just a single line showing that that neuron is talking and firing. Here, the threshold is this down here in the bottom graph. I'm just kind of like waving my mouse around. In this bottom graph down here, right, this would be the threshold, that fat red line. Here, the excitatory potentials, or the EPSPs, they outweighed the inhibitory ones, and it was enough to push us over threshold. With this one, we have the same amount of excitatory ones, but we have two inhibitory inputs here that are very strong. So we have more hyperpolarization happening. We don't reach threshold. So this top one is this guy, right? We fire action potentials, whereas if we now switched over to this second neuron here, where now all of a sudden we have more IPSPs, that will actually, as you can see here, it will silence the neuron and it will stop talking, meaning it'll stop firing. So you kind of see this elimination of action potentials because now we have some inhibitory inputs coming in. We don't reach threshold anymore. We don't fire an action potential anymore, right? So this could be a sequence of events where one neuron is talking and all of a sudden becomes silenced because other guys are inhibiting that neuron's ability to talk. And when I say talk, I mean fire an action potential. Okay, let's do a couple more. So the release of neurotransmitters. This is when, this right here, our terminal button, this is when the action potential comes down. And what I've been telling you guys is it triggers the release of neurotransmitters. Now there's a precise mechanism for that triggering process. So step one here, an arriving action potential, it depolarizes ion channels that are on the terminal button. And up until this point, we have not talked about ion channels on the terminal button. We've talked about ion channels along the axon, right? And on the dendrites and on the soma, but this is the first one and will be probably it will be the only one we talk about for the terminal button. Down here, this is the membrane of a terminal button. Whoa, didn't mean to do that. Calcium channel right here, right? When I say receptor, in this case, it's actually, take that back, not a receptor. When I say ion channel, you have ion channels across the entire membrane of the neuron. This one on the terminal button, calcium, if it gets depolarized, meaning we have an action potential coming in and it's strong enough to depolarize that ion channel, the ion channel opens up, just like sodium channels, right? When we talked about the action potential. The calcium channel opens up, allows calcium to enter the cell, right? So I was about to point on the screen. So if here is sitting, I'll wave my little thing around. If this is where the calcium ion channel is, calcium goes inside of the terminal button. When calcium enters, it allows this vesicle, this bubble that contains the neurotransmitters, it allows it to dock onto the terminal membrane and split open and dump all of the neurotransmitters into the synapse. So that's because calcium came in and allowed the proteins to bind from a vesicle to the membrane, right? The membrane has these little docking clamps, and the vesicle itself has some proteins on there. So when calcium comes in, it allows those two proteins to kind of dock, like a spaceship to a space station. They dock, and then they brutally tear it open, and the neurotransmitters fly out into space, meaning the synaptic cleft. So step 2, calcium ions that have entered the terminal button allow binding of proteins of presynaptic membrane to membranes of synaptic vesicles. Those proteins then are pulled apart and release the neurotransmitters. So the important thing to remember in terms of, I'll be more explicit about that in a second. The term to remember with these channels is that these calcium channels, just like the sodium channels of the action potential, are voltage gated, right? When we talked about the action potential, we reach threshold of excitation, sodium channels open and sodium flies in, right? The sodium channel was triggered by threshold, meaning it was triggered by a change in voltage. At a particular voltage, they opened up. At another voltage, plus 40, they close, they lock, and they become refractory. Same thing with these calcium ion channels. They open when they get depolarized enough, meaning the action potential traveled all the way down the axon, hit the terminal button, and at the terminal button, we have calcium channels, depolarizes, opens them up, calcium comes in and allows the vesicle to dock, open up, and dump all the neurotransmitters out. I need a sip of water. So these ion channels that are on dendrites, right? We just talked about ion channels on the terminal button up here. That's where the calcium ion channels are. But when we go here and we start talking about the ion channels on the postsynaptic membrane, meaning the dendrites or the soma, this is the postsynaptic membrane. This is where we're going to spend a little bit more time because we're going to talk about neurotransmitters, what kinds of receptors there are. I like to think of them, they're often in textbook illustrated as like they literally look like bell peppers. So keep that in mind, right? Just as a visual, what is happening here, the bell pepper has these little bumps here, and we call these binding sites. So when a neurotransmitter comes and it binds to it, there's a specific place for it, like a lock and key. Only a particular serotonin is going to fit on that particular binding site. When that happens, it opens up the middle, which is where the ion channel is, and allows ions to go through. So neurotransmitters do not go through the ion channel. Neurotransmitters open up the ion channel, right? It's going to be an ion like sodium, chloride, potassium, or calcium. Those are the 4, and that'll be on the next slide, or 2 slides from now. Those 4 ions are basically moving in and out of those receptors, meaning ion channels. Sorry. Receptors, in this case, ion channels, these are synonymous because these are all receptors or binding sites. All of these receptors are ion channels. So neurotransmitters binding with those receptors on the postsynaptic membrane. Again, the postsynaptic membrane is going to be dendrites or soma. It opens up ion channels, and it opens up neurotransmitter dependent ion channels because, again, a neurotransmitter, so here we have ions, this little black dude over here is the actual neurotransmitter. There's a binding site, it opens it up, and our big blue ions here can flow inside, or in some cases it might be something like potassium that actually leaves the cell. So the neurotransmitter determines what ion passes through. And we'll talk about them when we go through the different neurotransmitters. They're usually just very particular receptors, very particular ions that it's allowed to pass through. But that passing through, right, positive charge coming in or negative charge coming in, that's what changes the membrane potential, and that's what leads to IPSPs or EPSPs. So again, reminder, NT, I abbreviate neurotransmitter a lot to NT. So NT here means neurotransmitters. The neurotransmitter does not cross the membrane. It's the ions that are going in and out. Okay. Actually, I think I'm going to have to stop here. I was going to go a little bit further, but it's actually a pretty good stopping point. Because next we're going to talk about two different types of ion channels. We'll save that for next time. I'm not going to go into it right now. We'll stop there, talk a little bit more specifically about other events that are happening at the synapse. Because, for example, I told you guys neurotransmitters are there and then they get recycled or destroyed, so we need to talk about that. When we talk about, we're not doing psychopharmacology yet. We're going to do a little bit. But when you take, for example, an SSRI, serotonin reuptake inhibitor, which is an antidepressant, it's actually directly manipulating something happening at the synapse. That recycling process, it changes the recycling process for serotonin. So we'll talk about that more next class. But let me go ahead and stop there. How long was that recording? It was a 30-minute recording. So normally, right, with class, I would take a little bit slower. I would have us talk a little bit more about it. But because this is a recording, you guys can actually go back. You can go back and listen to it, watch it again. What else I was going to say? But at least it's an introduction to the basic terms, the terminology for the synapse. These are important. This way you can go back and review it. And I will also be uploading an online recorded lecture that I made for the action potential itself. So that I did that a year ago, but I'll upload that as well. It wasn't for a missed class that we had class, but that way, if you wanted some extra review for the action potential to also kind of like go back and listen to it, it could be helpful for those of you who want that. All right, I will see you in class next. Bye, guys.